Okay, well, bye. Okay, so hello everybody. I guess before I start, I'll make a sound check with Clotilde. Can you actually hear what I'm saying? You're gonna contact us somehow to let us know. Okay, so well, yeah, maybe I should slightly modify what Laetitia just said. So I do work on the immune response against polyomaviruses, which are persistent DNA viruses, but I'm not going to talk about polyomaviruses today. So I'm not really in my specialty. It's not the viruses that I work on or that I have worked on in the past particularly. Uh, okay, so is everything okay with Clotilde? Did she? Yeah, it's okay. It's good. Yeah. Yeah, but all I can see is Clotilde Philibert. That's all I can see. I can't see any chat or anything. Right. So what I'm going to be doing is, if I get back on this, is just give a bit of an introduction about persistent viral infections. And what is going to be important is the difference between chronic and latent infections, and then of the different types of persistent viral infections. I'm going to be talking about the immune response to chronic hepatitis, hepatitis B, hepatitis C, human papillomaviruses, and then herpes viruses. So I'm not going to talk about HIV, and I'm unfortunately not going to talk about polyomaviruses. So anyone who was who was in the lecture last year, I perhaps remember this slide here, which I copied from a textbook, and it basically gives you a summary of what happens after an infection. Either you manage to eliminate the pathogen, and that's a win for the host. You can have a progressive disease, and it becomes persistent, or you die. That's a win for the pathogen. And really, the way I see these persistent infections is it's kind of a draw, okay? You never really get rid of the pathogen, you never eliminate it, but you can, kind of be asymptomatic for a relatively long period of time or forever for some persistent viral infections. So it's a kind of like a standoff between the host immune response and the pathogen. So that's the kind of situation we're at here. So there are, of course, two types of persistent viral infections. There are latent and chronic infections. So both of them start with a primary infection, which, during which you'll have a peak of virus replication, followed by the manifestation of some symptoms. So in a lot of cases, it will be something not very specific, fatigue, tiredness, maybe some fever, and then you'll kind of get better. And for a latent infection, there will be no active virus replication during these periods of clinical remission. But the virus persists somewhere in your body in a latent form. And this latent infection can have or can undergo reactivations. So the virus will return to an active lytic replication cycle, and this will be accompanied by the reappearance of some kind of symptoms. And these cycles of latency and reactivation can go on for the rest of your life. And in contrast with that, you have chronic infections where there is always active replication of the virus, even in the absence of clinical symptoms. But these symptoms will tend to get worse over time. And in the end, can lead to organ failure and death. So I guess the classic examples of chronic viral infections will be hepatitis B, hepatitis C. They don't have any type of latency really. Maybe hepatitis B, but there's always a active virus replication. And the big specialists for latent infections are the herpes viruses, herpes simplex, varicella zoster virus, cytomegalovirus, and Epstein-Barr virus. And in between, you have things like HIV. So in an untreated patient, there'll be constant active virus replication, but it can't be cured because, or with antiretroviral agents in any case, because there is also a latent reservoir. So HIV is, chronic and latent, and papillomaviruses are a little bit like that as well. Okay, so the ones that are going to be, so really for all of these viruses, what is it that allows them to persist? I mean, we're talking about immunocompetent hosts here. We're not talking about people who have a defective immune system. So all of the affected people have mounted an antiviral immune response, but it's not sufficient to eliminate the virus. And that means that they've got to be able to evade the innate response, and particularly interferons, the cellular response, and the humoral adaptative response. And last thing I want to underline is, some of these persistent viral infections cause cancer. Okay, so papillomaviruses, the high-risk HPVs cause cervical cancer and some oropharyngeal cancers. HBV, HPC, HCV cause liver cancer, and Epstein-Barr virus, it causes several types of B-cell malignancy. So we're going to start off with the chronic hepatitis viruses, and their link with hepatocellular carcinoma is the main reason why they are a worldwide health problem. So, if you put both of them together, the estimate, I think this is maybe from a couple of years ago now, is that there might be, this is like 5% of the world population living with a chronic hepatitis B or hepatitis C infection. And the prevalence of these chronic infections is a pretty good match for the prevalence of hepatocellular carcinoma. Okay, so it's really these chronic viral infections, they're the major risk factor for developing these primary liver cancers. And that's why we want to try and control, and if possible, eradicate these viruses. So firstly, hepatitis C, and then hepatitis B. Don't know why, I somehow put them in reverse alphabetical order. So hepatitis C, that's 50 million people, roughly speaking, will have a chronic hepatitis C infection. In France, 130,000 people, so the prevalence rate is a bit low compared to the world. So the highest regions worldwide are going to be the Middle East and some North African countries, Egypt, Sudan, Libya. Yeah, that's right. So this number has gone down. So this image here was from maybe 5 years ago. The more recent data is like 50 million, estimated 50 million people. So there are 6 genotypes of hepatitis C, 1 to 6, with different subtypes, and they were identified and numbered in the States, in the USA. So genotype 1 is the most prevalent in the USA, then 2, then 3, and then so on. But in different regions of the world, this will not be true. Okay, so in Europe, maybe 2 is more prevalent than genotype 1. And in other regions of the world, there's different types of prevalence. What do we need to know about this virus? It's a member of the Flaviviridae family, but it's in its own genus called the Hepasivirus. So these are single-stranded plus strand RNA viruses with a genome about 10 kilobases long, icosahedral capsid with an envelope. Now, most of the transmission is parenteral, so via blood, either through iatrogenic transmission and also some limited mother-to-child transmission and some sexual transmission, but it's mostly blood transmitted. Tropism of the virus, hepatocytes, and maybe B lymphocytes. So if you get infected, then probably 80% of people will go on to develop a chronic infection, and only 20% of people will have spontaneous resolution. And if you've got chronic hepatitis C and you don't have access to antiviral medication, then you'll have a progressive disease with hepatic fibrosis, moving on to cirrhosis, and then finally hepatocellular carcinoma. Now, this is quite a slow process, and data from 20, 30 years ago showed that the median time from infection to cirrhosis was about 30 years. So it's something that happens over decades. And the cofactors being alcohol consumption, male sex, HIV co-infection, and age. So the older you were at the infection, the faster the progression was. Now, fortunately, it can now be cured because it's a huge advance in antiviral therapy over the last 15 years. So just a combination of 2 directly acting antivirals for 2 or 3 months will allow something like 95% of patients to have sustained virological response. That means there's no more viral RNA in the blood, and this persists after the end of the treatment. So that's good, can be cured. So basically we have the solution to hepatitis C now, it's the antiviral therapy. But these are kind of expensive. And so it'll be a combination of 2 different drugs on 2 different viral targets, okay? Okay, so just forget about therapy for now. So imagine the situation without access to therapy. This is a virus, it's replicating continually for 30 years in the person, and the individual can't eliminate the virus. So it's escaping from the innate response, the CTL response, and the humoral response. So how about the innate response? Well, there are 2 viral proteins that are involved here. So NS, these are the non-structural proteins. NS3, where is a protease, and it will cleave the cellular protein MAVS, which is an essential intermediate in RIG-I and MDA5 signaling. So all of the, the main detectors for intracellular detectors for viral RNA, their signaling pathway is being handicapped, being eliminated. Also, cleaves TRIF, which is a cofactor for TLR3 signaling. So all these RNA sensors, they're inactivated. And NS5A is an inhibitor of JAK-STAT signaling and also RNA-dependent protein kinase. So interferon signaling and the activation of interferon-dependent antiviral systems also knocked out by the virus. So these are the 2 main mechanisms by which hepatitis C evades the innate response. And the importance of NS5A is really shown by the clinical success of all these Tazvir drugs. Okay, so all these drugs that end in Tazvir, Daclatazvir, they're NS5A inhibitors. And if you look back in the literature, when these drugs were being developed, you had some phase 1 clinical trials just using these drugs as monotherapy. So you've got Daclatazvir here, used as monotherapy on genotype 1A or genotype 1B viruses. So genotype 1B viruses, look at this, just one drug, and you get like 1,000-fold reduction in viral load sustained over 14 weeks. So if you prevent the virus from doing this, then your innate immune response can like clear things up, okay, and control the replication. So this is, really a key mechanism for the virus that allows it to persist. Okay. So how does hepatitis C virus avoid the CTL response? Well, we know that the CTL response is important for resolution of the primary infection because there's a pretty good association between HLA alleles and whether you go on to develop a chronic infection. So CTL are important in clearance of the virus primary infection. But once you've got a chronic infection, then like with HIV, you've got this kind of like cat and mouse game between the virus and the immune system. So it's an RNA virus, it's got a high error rate, can produce a lot of variants every replicative cycle. And some of these can be CTL escape mutants. So it's just virus variability generating new escape mutants, and that's how your CTL response can't really keep up. It's not sufficient to eradicate the virus. Once a chronic infection has been, started. And of course, it's kind of dependent on what your HLA alleles you have. If you've got the right HLA allele that's going to present a conserved epitope, then the virus will find it more difficult to mutate and avoid the CTL response. If you're unlucky, you've got the wrong HLA, well, the virus will find it easy to mutate those epitopes. Okay, how about the humoral response? Similar kind of thing. Firstly, the virus can mutate the epitopes on its E1, E2 surface glycoprotein, envelope glycoprotein. So the epitopes can be modified, but there's something else which is perhaps as important or more important is this thing that's called the glycan shield. So there are a lot of glycosylation sites on the E1, E2 dimer. So you've got like 5 on E1 up to 11 on E2. And here you can see that the glycans here, they're kind of forming a protective shield that's around the neutralizing epitope. So it makes it difficult for neutralizing antibodies to get access to these sites that are very, very important for the interaction of the virus with its receptors. And one of the problems is you can also have this thing called the glycan shift, which means that these glycosylation sites, so they're all N-linked, so they depend on these asparagine residues somewhere on the glycoprotein. And if these asparagine residues mutate, then instead of having a glycan on this residue here, you can get the glycosylation, which will start on another residue. So this glycan shield isn't a static thing. It can move. You can have different variants that are selected. So this makes it very difficult for people with a chronic infection to get an effective neutralizing response against the virus variation. So it's a similar kind of thing with HIV again. Glycan shield, glycan shift is a concept that comes from HIV. The other thing that hepatitis C can do is that it doesn't actually need to be released as a free virus or associated with the lipid transport proteins, but it can also spread cell to cell. So cell to cell spread is generally resistant to neutralizing antibodies. And that's true for many viruses, but also for hepatitis C. So here's some data showing hepatitis C virus being transmitted in the culture either as a free virus, that's in the black bars here, or with cell-to-cell transmission in the presence of increasing quantities of 2 different monoclonal antibodies. So with the black bars, you can see that this monoclonal antibody here, okay, 10 microgram per ml, is really blocking all the transmission of the free virus, but cell-to-cell transmission still can occur. Okay, so if you've got a high titer of neutralizing antibody in the serum, it's not going to block all of the transmission events because the cell-to-cell transmission can still get through. So that's hepatitis C. What about hepatitis B? Okay, so hepatitis B, maybe 5 times as many people with a chronic infection. Now, that's a significant proportion of the world population, 3% of the world population. France, well, not so many, 10 times less. And the main areas for hepatitis B antigenemia is going to be in the East Asia region. Okay. Maybe sub-Saharan Africa as well. So just like hepatitis C, there are multiple genotypes and subtypes with different subtypes and genotypes prevalent in different areas. So in Europe, we've got A2, D, and A, D, and G. East Asia, they have some specific genotypes like I. In Africa, they've got E. We don't really have E in Europe. Now, what about the virus? What is it like? Okay, so it's part of the Hepadnaviridae. It's got a small particles, small envelope particles, small circular genome, partially double-stranded. Silent mode, thank you. And it's interesting because it's a reverse transcribing virus. So the DNA inside the virus particle is transported to the nucleus, but then it's going to be expressed as RNA, which gets reverse transcribed back into DNA inside the particle before the virus is released. And transmission, once again, is like blood-borne, with major modes of transmission being sexual transmission and mother-to-child. So mother-to-child transmission during birth, not in utero, during birth, is probably the main motor of HPV persistence worldwide. Okay. Once it gets in the blood, it's going to affect hepatocytes. It doesn't really have any other extrahepatic site of replication. And just like hepatitis C, if you have a chronic infection, it's going to develop over several decades to give you cirrhosis and hepatocellular carcinoma. So I suppose the good news, if you're an adult, is that if you get infected with hepatitis B, probably you'll have about 80% chance of spontaneously eliminating the virus. Only 20% or maybe 15% of these infections will become chronic. Whereas if you're infected on the day of your birth as a baby, then the proportions are reversed. Okay, so most of these perinatal infections will become chronic, maybe 80% of them. So that's why this is really the big and the most important mechanism of transmission. And for development of cancer after if you have a chronic hepatitis B infection, cofactors are once again age, male sex, alcohol, and HIV co-infection. All of these things will make it worse. Okay, how are these treated? So we do have antivirals that are effective against hepatitis B virus. The virus has only got one enzyme, which is reverse transcriptase. And you either get entecavir or tenofovir. So if you've got HIV co-infection, you probably get tenofovir because that will be part of your HIV regimen anyway. These therapies, they can control or reduce the level of virus replication, but they're not curative. So it's like HIV, you have to keep on therapy for long term. There's no real advantage in combination therapy. It's been tried, and there isn't any other pharmacological target. So you just have monotherapy. So that's the situation. So unlike hepatitis C, there's no curative antiviral therapy. Um, yeah, and that's really one of the big challenges for hepatitis B research. Okay, so how does hepatitis B avoid the innate immune response? Well, some of the things we know about this come from experimental infections in chimpanzees. Now, to be honest with you, I personally think this type of research should not have been done for ethical reasons. I think a chimpanzee is just about the same as a human being. I don't think we would do this with a human being. We shouldn't have ever performed this kind of experiment. But on the other hand, there's a scientific part of me that thinks, oh yeah, this is really interesting, what's happening. So there was work done, in the 1990s, early 2000s, where there was a experimental infection of chimpanzees by hepatitis B virus. And once you do that, what you can do is you can, the time of the infection, you can follow a different number of markers. So you've got hepatitis B DNA, which is in the kind of diamonds here. And you've got peak of virus replication, but it's not really accompanied by liver damage. So that's the limit. Liver damage is here is, what's that, adenine transferase or something like that, okay, so some liver enzyme. So the peak of virus replication isn't really accompanied by the peak of liver damage here, okay? And if you look at the induction and there were liver biopsies taken at different times after the infection and analyzed by northern blot, really kind of old-fashioned technique here, but it allows you to see at this time point, 11 weeks to 15 weeks, that's when you get an infiltration of T cells, okay? You've got CD3, you've got interferon gamma that's coming here. The T cell responses are coming up at, 4 weeks post-infection. And if you look at the engagement of the interferon response, where this is OAS, so this is 3'5' oligoadenylate synthase, one of the big interferon-induced genes. Right at the peak of virus replication, you don't really get very much. Okay, OAS is staying quite constant, maybe a little bit here, but it comes up when you have the T cell infiltrate. Okay, so what does this mean? This means that if you get a primary infection, you've got a peak of virus replication, but it's your innate immune system doesn't notice anything. Okay, no, just like it's as if nothing is happening. So it's a stealth infection. Now, one of the questions you can ask about this is whether it's just your innate immune system in hepatocytes doesn't know the virus is there, or whether the virus is actively suppressing the innate immune response. And this was done in this kind of experiment I think is really great actually. So they got liver biopsies from a patient with chronic hepatitis B or controls, non-infected. And they put them in culture with or without poly-IC, which is a good inducer of interferon responses. So, and they looked at the mRNA expression for interferon beta. So this is what we got here. So this is the control condition. So no stimulation. So the non-infected people, hepatitis B infected people, they've got no expression of interferon beta mRNA. Okay. Even if you're infected, your level of interferon beta mRNA, nothing, just the same as the controls. If you add poly-IC, that's this condition here. Okay, in the non-infected people, you get interferon beta mRNA expression, then it goes down. But with the HBV infected livers, just the same. Okay, so there's no inhibition of interferon induction here. Okay, so the virus isn't doing something to suppress the interferon response. It's just like not being detected. Okay. I'm going to tell you about this because I kind of remember it because it's so fun, right? So how do you do this kind of experiment, right? So they get the liver biopsy, it's in a big needle, and they chop it up into little cylinders. So each one they put it in cell culture medium and they add stuff and they take a bit out. They analyze one at 0 hours, one at 3, and one at 6 hours. And this was done with within 3 hours after the liver biopsy. So it was quite tricky to do experimentally, but I thought very good. Okay, so why doesn't the virus get detected here? So I think it's really to do with the replication cycle. So one of the, well, perhaps the main way viruses get detected is by detecting unusual types of nucleic acid in the wrong compartment in the cell. But how does hepatitis B replicate? It's going to infect cells. The capsid will probably dock with the nuclear entry pores, and you only get the virus DNA delivered directly into the nucleus. So this looks like normal cellular DNA. Then the viral RNA is expressed here, just like a normal messenger RNA. It's going to be exported. And when the virus replicates, the reverse transcriptase will synthesize the DNA inside the capsid. So it's not actually out in the cytoplasm. It's not accessible to Rig-I or CGAS or something, or any of these nucleic acid receptors that can detect unusual RNA or DNA structures. So I think somehow this virus has found a way to, replicate, hide its different types of nucleic acid in the right part of the compartment and not be detected. Okay, so escapes from the innate response, doesn't get detected. However, people do develop an adaptive response, CTL response and a humoral response. And whether you eliminate the virus depends really on whether you have a full seroconversion or not. So people who have had an acute hepatitis B infection and eliminate the virus will have full seroconversion. So they'll develop antibodies against the surface glycoprotein, the S antigen. So you'll be anti-HBs positive. Then you'll develop antibodies against the capsid antigen, the HBC antigen, and against the rather mysterious HBe antigen. And of course, because you have the antibodies, these two antigens won't be free in the serum or the plasma. However, if you have chronic aggressive hepatitis B, you won't get seroconversion against these two antigens, and the two antigens will be present in the blood. And if you have persistent HBV antigenemia, you get, once again, partial seroconversion. You develop antibodies against HBe. You don't have any HBe antigen circulating. Now, the patients who are at high risk for hepatocellular carcinoma are these ones, people who are negative for anti-HBs antibodies and negative for anti-HBe. So what does that mean? That means that if you get a good seroconversion against HBs, that's it, you eliminate the virus. And this is really the basis of the vaccination, because the vaccine is a recombinant HBs antigen. Okay, so you might say, okay, we've got the vaccine, why don't we just vaccinate the people with the chronic aggressive hepatitis B, give them that vaccine, they'll get the antibodies and they'll be all right. Doesn't work, okay? They're resistant to this, to the vaccine. So I think that means that there's some kind of antigen-specific tolerance against the HBs antigen. And why does this occur? Well, one of the things that is quite particular and specific to hepatitis B is that when it replicates in hepatocytes, it doesn't just release infectious particles. It releases a mix of infectious particles that are in the minority and a lot of empty non-infectious particles. So if you look at this electron micrograph here, these ones here, these are the full capsids, right? They've got virus DNA in the middle. They've got the core antigen. These things here, these are just like small envelope structures with the HBS antigen. And it seems like what your immune system is seeing is this one. When you get a partial seroconversion, you seroconvert against the core antigen, HBC antigen, and it's ignoring all of these ones. This one's got DNA in it, right? So your antigen presenting cells somewhere in a draining lymph node will get activated. They'll be able to present these antigens. All of these empty particles, they'll probably be tolerogenic. And there's a lot of them, okay? So it's a kind of like high dose tolerance sort of thing. So that's the main mechanism of avoiding the humoral response, is like somehow finding a way that your host immune response just thinks it's not there, that it's not dangerous. Now, one of the things that hepatitis B does not have is a lot of variability, doesn't have a lot of antigenic variability. And that's because the virus has got to code for the P, this is the polymerase, the capsid antigen, and the surface glycoprotein on a very, very small genome. And so the surface glycoprotein, HBS antigen, is coded on the same part of DNA as the polymerase, but in a different reading frame. So that means the sequence is highly constrained. Okay, because anytime you get a mutation in the hepatitis HBS antigen, it's going to have an impact on the polymerase at the same time. And the virus has got to have both of these proteins that retain their function. So that means it can't really escape from neutralizing antibodies, and it can't really, there's not much serological variability between genotypes. So the same vaccine is effective worldwide, irrespective of the hepatitis B genotype. That's also probably the reason why antiviral monotherapy can work, because the polymerase is also under the same type of evolutionary constraint. Okay. Now, how about the T cell response? Several people have looked at CTL responses against epitopes in different virus proteins in people with acute and chronic hepatitis B infection, and most of the adults with an acute infection will eliminate the virus. Okay, so what you see in an acute infection is going to be an effective antiviral response. So I better hurry up actually. So in acute infection, you can see, quite a reasonable number of these HBV-specific CTL, and in a chronic infection, really, really a lot less or not detectable. Okay, so there are several studies that show that you've got reduced numbers of HBV-specific CTL in chronic infection, and especially for the HBS antigen. Okay, where you don't get the seroconversion, there's almost no HBS antigen-specific CTL either. So why is it that the hepatitis B is avoiding the T cell response? Well, part of it is that people with a medical background, I guess, know this better than me, but there's something a bit unusual about the circulation in the liver, in that you have this kind of what's called the space of Dis here, and the hepatocytes can send some of their protrusions across it and interact with lymphocytes directly. So antigens presented on these on the HLA molecules of an infected hepatocyte can really be presented directly to a T cell that's infiltrating into these into the sinusoids in the liver. So you can get this kind of antigen presentation that occurs without an innate danger signal, because we've seen that inside the infected hepatocytes there's no interferon activation. So you have this situation where the specific T cells might be migrating here and interacting with epitopes presented on infected hepatocytes from the virus, and this could be more tolerogenic than immunogenic. Okay. So how can we get any kind of effector T cells produced at all? Well, this is probably happening in the secondary lymphoid organs, okay, but not directly in the liver. Okay, so one of the questions is how do we, so the antiviral therapy can't cure the infection. And this is one of the, and we know that if patients seroconvert, then they can eliminate the virus. So this is really one of the main reason why a lot of, for maybe 20 years or more, people have been trying to look for immunotherapies that can reactivate the defective antiviral immunity, induce seroconversion. So I had a bit of a roundup of the studies last year, and I looked quickly this year. I didn't really find anything new. So there's three different kind of approaches. One of them is, okay, so we know that the innate response is defective in the infected hepatocytes. Let's try giving a TLR agonist, boost the innate response. So this has been tried in phase kind of 1-2 trial. 39 patients got the TLR8 agonist, 9 patients got placebo, and you got clearance of the virus, 2 out of 39 patients in the treatment arm. Another thing you could do, checkpoint inhibitor, trying to combine this, and in both cases combined with nucleotide inhibitors, so some kind of antiviral therapy. Okay, trying to give these anti-PD-L1 checkpoint inhibitor antibodies, 6 months of therapy, this time low dose group, high dose group. And the only thing that is slightly encouraging is when the baseline viral load is low, there seem to have some people who can clear infection. But for high viral load, not very encouraging. And last thing you can try and do is have a therapeutic vaccine. Okay, so the hepatitis B vaccine is recombinant protein with adjuvant, and we know that doesn't work. So the idea is let's try and give some different kind of vaccine formulation and see if that works better. So this was a study testing viral vectors, modified virus ankara, and recombinant adenovirus, chimpanzee adenovirus, either on its own or 2 doses with or without checkpoint inhibitor. So a lot of different trial conditions, really only 2 out of 55 of the patients managed to clear their virus. So we're not really there yet, okay? That's about the situation. There's no real cure for hepatitis B and nothing that looks really fantastic either so far, I have to say. Okay, so that means that if we really want to eliminate hepatitis B, then we have to think ahead. So when are the most transmission? We have to block transmission. Okay, if we block transmission, we don't have people with chronic hepatitis B. The health problem will be resolved. And really, since worldwide the major mode of transmission is perinatal. Okay, so the recommendations is that you should vaccinate babies on the day of their birth, then have a boost at 1 month and then at 6 months. And so these infants who get vaccinated will seroconvert, and that will prevent chronic infection. So this is really, I think, quite interesting because it means that, okay, this baby that's getting infected and getting vaccinated on the same day. Okay, so you've got a kind of a race between your immune system and the virus. And the virus is going to try and install this chronic infection, and the vaccine is going to try and induce the antigen-specific humoral response. And in most cases, just giving the vaccine on the same day as the infection is enough. Your immune system gets enough of a head start to be able to eliminate the virus. How do we know this? Actually, the best data I found was from China, because they've got like cohorts of a lot of people followed for several decades now. So they had a randomized controlled cluster randomized trial, which was carried out in one of the provinces in China over 5 years. And they now randomized 40,000 newborn babies, right? Vaccine or no vaccine on the day of birth, and then boosters. And they followed up over like 10 years and 20 years later on. Okay, so this is in, okay, this is the no vaccine group. They had like 9% and 6.7% antigenemia at 10 years or 20 years after birth. In the vaccine group, it goes down to 22%, 1.8%. So like 80% effective or something like that. And they also have results from the liver cancer, okay? So you can see in the control arm here, now we've got like 30 years follow-up on this cohort. And you see there's some people getting liver cancer at 20 years old, 25, because of their hepatitis B infection. And that's pretty much all stopped here because of the vaccination. So this is really telling us that, and currently, that hepatitis B S antigenemia in children under 5 years is down to 0.3% in China because of their vaccination drive. So they've reduced their disease burden in the future by about 97% because of this. And if we do this worldwide and we keep it going, that's it. It's finished for hepatitis B. That'd be a great win for humanity. I like biodiversity, but I make an exception for viruses. Okay, so but maybe not in the USA. I don't know if anybody's been following this story, but Robert F. Kennedy Jr. has been put at the head of the US health services, and he recently, this summer or last summer, replaced all the members of the Advisory Committee on Immunization Practices, which is a CDC committee. So he put a bunch of new people in there, and they started to think about, shall we delay the birth vaccine dose? So today they say that they want to shift and vaccinate only from 1 month only. And then the data we have shows us that this is already too late, or it's less effective, okay? Like I said, it's a race between the virus and your immune system. You're giving the virus a 1 month head start here. Okay, so one of the arguments in the States is, yeah, but we only need to have the birth dose for women who are chronically infected, the pregnant mothers. But in the States, we've got 7% of live births are to women who don't have any prenatal care or inadequate prenatal care. So if you've got screening for everybody, okay, that makes sense. But if you've got a large part of your population who has no prenatal screening, you don't know their HBsAg antigen status. So all of these women, those who are chronically infected, if you delay the vaccination, that's going to be definitely a loss for public health and humanity. Okay, so we got really not so much time. I'll have to try and go a bit quicker. So so we leave hepatitis and go on to papillomaviruses, which, as you know, is the cause of cervical cancer. This is the fourth leading cause of cancer in women in 2022, apparently, causing about 350,000 deaths worldwide per year. And it's exclusively caused by high-risk human papillomaviruses. So there are hundreds of different papillomaviruses that can infect us. Most of them are entirely harmless. And by their sequence homology, you can group them into alpha, beta, and gamma groups. And it's only the alpha group that contains high-risk HPV. So, and even within alpha, not all of the alpha HPVs are high risk, but all of the high risk HPVs are alpha. Okay, if everybody understands that. So these infections, papillomaviruses, small double-stranded circular DNA viruses with a non-enveloped icosahedral capsid. For the high-risk HPVs, sexual transmission, they have tropism for the basal epithelial cells. So the disease progression is that you'll have primary infection, and this can progress to low-grade lesions, high-grade dysplasia, and then finally invasive cervical cancer over several years, maybe decades. And the progression from infection to cancer is low penetrance, so that at every stage, there is a high chance that the infected woman will be able to eliminate the virus. Okay, so even if you've got like a low-grade lesion, just left on its own, most of these would clear up. Okay. Now, what I said about the HPV alphas, there are a limited number of them which cause most cases of cervical cancer. So the most dangerous one is HPV 16, and then all the others, HPV 15 is second. Okay. But these are not the ones that you find mostly in women with a cytologically normal cervical sample. Okay, so what about progression to cancer? What are the risk factors if you've got a positive smear test? Cofactors are number of full-term pregnancies. I don't know why this is. Tobacco, cofactor, and HIV co-infection. These are going to be bad for you. And then what's going to be the treatment? Well, if you've got a low-grade lesion, just like do nothing, most of the time it will clear up on its own. Just needs to be like followed up. If you've got, so SIN is cervical intraepithelial neoplasia, low grade, and then higher grade SIN2, SIN3. These have got to be treated by removing the lesion physically, conization or with cryotherapy, and this is some large loop, some kind of surgical thing. Okay, don't ask me what this is. It's too many letters to fit on the slide. And if you've got, actual cancer here, invasive cervical cancer, well, then depends on the stage and what the protocols are. So in these cases, okay, so most of the time you can actually eliminate these viruses. It won't go on to a kind of chronic infection that will lead to cancer. But in some cases it does. So why can the virus manage to do this? Well, it's something to do with its life cycle, its replication cycle, because it's going to be infecting these basal epithelial cells, but it won't have very much active replication. You'll just get maintenance of the virus genome at a relatively small number of copies per cell. You only get expression of mainly the E1 and E2 proteins and maybe E6 and E7 that are going to drive this low-level genome replication. And then as these cells divide and differentiate and move up through the epithelial cell layer, then you get more virus genes that are expressed. You'll get E4, which causes the amplification of the viral DNA. And then as the cells get right to the top layer, then you finally get the structural proteins, L1 and L2, that form the capsid. So you only get high-level virus replication and expression of viral proteins up here in the epithelial layer. And if you don't have any inflammation, then this is kind of an immune privileged site. You don't really have a lot of immune surveillance going on up here. Okay, so you don't really have active replication down here, which is more accessible to infiltrating leukocytes. So that's, I think, why the virus can go undetected. But of course, this depends on, virus replication not inducing an interferon response in the infected cells, or even down here. So how does that work? Well, it depends on TLR9, because HPV 16 infected epithelial cells really reduce the expression of TLR9 mRNA. So this was done about 10 years ago. You've got non-treated cells and then cells infected with pseudotype virus. This is just particles, but they don't replicate. QSV, this is quasi-virus particles, they do replicate, and they are treated with UV radiation to prevent replication. So if you've got a replicated virus, you can't detect any TLR9 mRNA anymore. And this is also true in biopsies. Okay, so if you've got a histologically normal tissue here, then you can detect TLR9 expressed in the cervical epithelium. In a lesion with HPV 16 infection, nothing. Okay, turns off TLR9, so it eliminates the detection of viral DNA by the infected cells. And other information that this is important is that there's a pretty good correlation or several correlations between TLR9 polymorphisms and different types of outcome, early stage cancer, persistent infection, HPV-16 copy number. And rather interestingly is that bats also carry a lot of papillomaviruses, and there seems to be a co-evolution of the bat virus genome and the TLR9 so the host TLR9 and the bat genome they are caught in this kind of evolutionary arms race to detect the pathogen or avoid detection. So this is the main mechanism for HPVs and perhaps other papillomaviruses to escape the innate immune response. Okay, for the adaptive response, well, we have effective vaccines. So the vaccines are formed of just empty capsids formed of the major capsid protein L1. So if you vaccinate somebody, you get antibodies against the L1 protein. And well, the first two were the bivalent and tetravalent vaccines. And the bivalent and they're both inducing antibodies against HPV-16 and HPV-18. The tetravalent also had HPV-6 and HPV-11, which are not associated with cancer, but are associated with genital warts. So the red arrows here, or red lines here, these are the antibody titers against the different HPV genotypes. So basically, you get antibodies against one genotype and you don't get very much cross protection. You get a little bit of cross seroconversion with HPV 31 and 45. But basically, you get antibodies against the vaccinating genotype and not very much else. Now, one of the things everybody should be clear about is that the antibodies protect against the infection, the primary infection. They won't protect you against the progression of the precancerous lesions, because once you get to this cancer stage here, then you lose the expression of the structural genes. You keep the expression of E6 and E7, which are the viral oncogenes that are pushing the cells to a cancerous transformation. So you don't have the expression of any of these genes anymore in the cancer cells. Okay, you have E6 and E7 only. And the virus can either be persistent as a kind of partial genome integrated into the chromosome or as an episome. Okay. So how are we going to activate, or can we well, okay, so we've had the innate response, we've had the humoral response. How about the T cell response? So over a period from about 1995 to 2005, there were a few studies, small number of women at each time, but overall you could find that the women who managed to control their infection, they actually have a CTL response against these virus oncogenes, E6 and E7. And the women who progress, who have a higher grade lesion, they have lower or undetectable CTL against E6 and maybe also E7, okay? So these are the virus oncogenes that are present in the cancer cell. So we've known this for about 25 years, and ever since like the year 2000, people have been thinking, okay, if we can induce the CTL against these targets, then we can get an anti-cancer CTL response, which will prevent the progression of the lesion, and prevent these cancer deaths. So there have been a number of, the first clinical trial I found was from 1998, no clinical response. And then people have been trying to use different types of, vector to get an effective CTL response. And the timeline for this, if you're an academic doing something like this, so Toos Damen in Holland, this was because the idea was here, right? Published in 2000. She managed to get the clinical trial run and reported 2021. You've got like a 20 years gap between your initial concept and the clinical trial. So we have perhaps the best result was in 2015. Okay, published 10 years ago, which in the abstract they said they had the first therapeutic vaccine to show efficacy against high-grade lesions associated with HPV 16 or 18. So let's have a look at this study. So it's a randomized controlled trial for women with SIN2, SIN3 lesions. You had a group who got the DNA vaccine versus placebo, and the endpoint was regression of the lesion to a lower histological grade. And so in the treatment group, they had like 55 women had a regression out of 114, and in the placebo group, only 12 out of 40. And this was statistically significant, and also in the treatment group, there are more women who cleared the virus DNA. So seems like it's working, right? So why hasn't therapeutic vaccination been adopted? So this was 10 years ago, we should be doing this by now, right? Well, probably not, because when you look at the efficacy, it's about, you can have regression in about 50% of cases. And that's only about 50% higher than in spontaneous regression, whereas, the surgical cure rate is more than 80%. So that's why therapeutic vaccination hasn't been adopted yet, because the surgery is much more efficient and much better for preventing lesion progression. Okay, so the only thing that's kind of hopeful about this is that when they looked at the women who had the vaccine and they had a good clinical response, their CTL response was higher than the women who didn't have a good clinical response. So maybe there's some future in this idea, and if we could get a better vaccine, then that could give you a better clinical response rate. But I haven't seen anything this year to really changes things in that respect. Okay, so we got about 25 minutes to think about herpes viruses. Well, I guess should be okay. So herpes viruses, so up to now, the viruses we've been thinking about have been quite simple with a simple genome. Herpes viruses are annoyingly complex. So they're big envelope viruses with icosahedral capsid with big complex genomes. And all of them are capable of a latent infection. So they can infect cells, have a lytic replication cycle, or have a latent infection cycle where there's only limited gene expression. Now, among human herpes viruses, the orthoherpesviridae family, they can be classed once again into alpha, beta, and gamma herpes viruses. And the alpha herpes viruses are herpes simplex 1 and 2 and varicella zoster virus. And in the beta herpes viruses, the one that's going to be interesting for us is cytomegalovirus CMV. And the gamma herpes viruses, Epstein-Barr virus and Kaposi sarcoma herpes virus, EBV and KSHV. So maybe about 10 years ago, these became the official abbreviations and the names, like human herpes virus 1, 2, 3, 4, 5, 6, and so on. These are no longer the official names, but people still use both the older name and these ones. So only the gamma herpes viruses are associated with cancer, but both the alpha and the beta herpes viruses can be problematic. So just one word about genome structure. So all of them have got like terminal repeated sequences at the end of their linear double-stranded DNA genome. And in the alpha and the beta herpes viruses, you've got terminal repeats on the long segment and terminal repeats on the short segment. So the internal repeat short, terminal repeat short, internal repeat long, terminal repeat long. And this is what defines what is called the unique long and the unique short region of alpha and beta herpes virus genomes. So all of the genes in HSV, in CMV, they've got names like UL something or US something. Is because of their place on this part of the genome or on this part. Whereas all of the EBV genes, they've got crazy names that have nothing to do with these ones. Okay, so we're going to be talking about UL, US from now on, and you should, just so you know, where this terminology comes from. Okay, so I'll talk a little bit about pathology and the health problems associated with these, well, maybe not all of them, but some alpha, beta, and gamma herpes viruses. So for the alpha herpes viruses, for herpes simplex virus, HSV1 causes oral herpes. Probably 70% of people have been infected with this virus in the room. It remains latent in neurons. So, the initial lesion caused by the lytic infection is around here. So the neurons that are going to be infected primarily are going to be in the facial nerve and the trigeminal nerve up here somewhere. And you can get recurrent lesions due to reactivation of lytic replication provoked by stress. So that's rather annoying, but it's not such a big deal. The big deal is HSV encephalitis. So this is very, very rare, and it's can be devastating if a child has a HSV encephalitis. So there is an antiviral therapy, which is acyclovir. So varicella zoster virus causes childhood disease, la varicelle or chickenpox. Probably everybody is infected as a child in France. I know certainly my kids all were. And I didn't get infected myself, so I guess I must be immune, so I must have had it as well, right? Once again, there's some neurotropism. So you get latent infection in neurons that can be reactivated in older adults. And this will give you a zona called shingles in English. Can be very, very painful on different parts of the body. So these can be treated by antiviral drugs. And because the frequency of reactivation is higher in older adults, then the current recommendations are, I guess, for older adults to get vaccinated, because you can reboost your antiviral immunity. I guess, anybody know this? Probably the recommendation is Shingrix, right? Not for the older one, the Zostavax, because it's more effective, more efficient. So this was the older vaccine, and this is the current one. So depending on the country, routine childhood vaccination is recommended. Like in the States, they recommend to vaccinate against VZV. It's not in the childhood vaccination schedule in France, right? Oh, thank you so much. Yeah, so that's kind of interesting. So I guess here we think, it's basically quite mild primary infection. There's actually not much of an individual benefit for the child to get vaccinated that would justify, the cost and of the vaccination. Whereas in the States, and other countries, they do vaccinate. Okay, so, oh yeah, the other thing I'm not going to talk about, but there's also increasing evidence that these reactivation could be related to the development of neurodegenerative diseases later in life. So this is another argument for generalizing vaccination in older people. So yeah, I'm didn't read up on this, but there's some indications that it could be a cofactor for neurodegenerative diseases. Okay, CMV, beta herpes virus. What is the problem here? Once again, primary infection in childhood, you get flu-like mononucleosis syndrome. Here, the latent infection is in myeloid progenitor cells in the bone marrow. So it's not in neurons, which is will cause a different problem. So everybody or people who've been infected, seroprevalence 30 to 50% is in France, not worldwide. Worldwide, it would be higher. And in the west of France, I believe we're pretty low. Don't know why, more like 30%. So people who have a latent infection are perfectly healthy, walking around, living their lives. But you can have reactivation, which can be very problematic if you have immunosuppression. So clinical AIDS, not really seen very much anymore, or if you have a solid organ transplant or hematopoietic stem cell transplant. So if you're under immunosuppressive therapy because of the transplant, then this can either reactivate your autologous CMV. If you've got a hematopoietic stem cell transplant, the danger is that the cells that you get grafted will carry the virus. So the highest risk association is a donor seropositive and a receiver seronegative, because you get a primary infection when you have no functioning immune system. So the other concerning clinical situation is when you have a primary infection in a pregnant woman, because you can have maternal fetal transmission in utero. And this can cause, deafness, probably the first cause still of congenital deafness in France. You can have very severe neuromotor problems or even death just after birth. And the earlier the infection is, the more severe the symptoms are going to be at the birth of the child. So this is a real problem here. So Epstein-Barr virus, once again, primary infection in childhood or in young adults. If you're a young adult, you'll get infectious mononucleosis. And then it will remain as a latent infection in B cells. So reactivation of these latent infections, there's some kind of epidemiological link between flares of autoimmune diseases, maybe something to do with long COVID. But also the main thing is that EBV is related to several B cell malignancies and also nasopharyngeal carcinoma, perhaps in Eastern Asia, and all 10% of gastric cancers, which is, I guess, less known. I really don't know why. No real antivirals for this one. Okay, so all these viruses, they're escaping, they're persisting, they despite being infecting people with a functioning immune response. So they have big, complicated genomes with a bunch of genes that can do lots of things to screw up your immune system. And of course, they are all messing around with activation of your innate immune response. So herpes simplex, a CMV, they have proteins that will, oh, hang on. I'm saying things out of order here. I didn't practice enough. Okay, so this is the part where I say, how do your cells detect infection? This wrong, okay? So you've got part of your, detector systems for DNA. Some of them can be activated by herpes virus infection. Rather more surprisingly, the innate immune receptors for RNA can also be important. So toll-like receptor 3, we know it's important for the control of herpes simplex HSV1 infection, because the deficiency, the innate deficiency in TLR3 is associated with HSV encephalitis. Also, RIG-I seems to be important as a mechanism of detecting herpes virus infection. These two things always confuse me. I never really found a good mechanistic explanation of why TLR3 is so important for this DNA virus. Okay, I really don't know why this is. For RIG-I, we actually know why. Okay, so it's quite interesting because what seems to be important here is that the the infected host cell is not detecting a viral RNA, it's detecting this RNA here, which is a 5S ribosomal RNA pseudogene, host RNA, which normally will be kept in the nucleus, and if it gets out into the cytoplasm, will bind to some host RNA 5SP binding proteins. Now, as part of the virus infection program, it's going to shut down the expression of host mRNAs and convert the ribosomes to expressing the viral RNAs. And part of this is inactivating the host genes that will bind to this RNA. So if the virus shuts down the host mRNA translation, then you get the increase of this RNA in the cytoplasm, which is what's going to activate RIG-I. So you've got this clever system in the host, which is not actually detecting a viral RNA or viral DNA. It's just detecting a virus screwing around with ribosomal translation. So that's pretty clever. So that's why this one works. And finally, TLR2 can detect apparently the highly hydrophobic patches on some of the herpes virus glycoproteins. Okay, so now I should be explaining how the virus messes around with all of this. Okay, so basically, one of the structural things that herpes viruses have is you've got the envelope glycoproteins here, you've got the capsid there, and in between them, they've got a bunch of things called the tegument proteins. So when the virus infects a cell, you have fusion of the envelope with the host cell membrane. And that means all of these tegument proteins get released into the cytoplasm before you have any expression of viral genes. And these can really mess around with the host cell innate immune detectors. So you've got a bunch of tegument proteins which will interact with and target IFI16 for degradation. It's an important receptor for viral DNA. Similarly, you have different tegument proteins inhibiting C-gas sting pathway, inhibiting the interferon response factor 3, the major transcription factor, and other proteins inhibiting RIG-I and C-gas. So you've got these tegument proteins which are going to be inactivating the host innate response. Okay, well, we've got 10 minutes, so time's getting a bit short here. What am I going to have to talk about this? Yeah, I'll probably just talk about CMV here. Okay. So how many slides do I have? Okay, I'll probably just talk about CMV. Okay, but yeah, let's just talk about CMV. So what are the protective responses that allow you to control the primary lytic infection? It's the CTL response against the first viral proteins that get into the infected cell. So these will be the tegument proteins like CMV, PP65, or the capsid proteins. They get delivered into the infected cell. If you've got a good CTL response against these proteins, you can eliminate the infected cell before it starts to express virus genes. Or they can also be the first viral genes expressed. So herpes viruses, during the lytic infection, they will express what are called the immediate early genes, then the early genes, then the late genes. And an effective CTL response will be targeting the first ones that are expressed, which allow the virus to activate its lytic replication cycle. So if you've got a latent infection, most of these T cell antigens are not expressed. Okay, So that's how the virus will hide from your immune response, the CTL response. But as these viruses are going to reactivate, they'll start off re-expressing these immediate early genes, and this will re-stimulate your memory T cells. So as you've got like these persistent or reactivations of the latent virus, you've always got your T cells being re-stimulated with the same antigens if you are seropositive for these viruses. So that means that if you're seropositive, then you end up having a pretty high proportion of your circulating T cells specific for these immunodominant antigens that are expressed early in the virus replication cycle. And when you're young and healthy, that's fine. But as you get older, these T cell responses, become a little bit senescent, and that's why you can get, these more symptomatic reactivations as you get older, because your T cells, they're just like, they've run out of telomeres or something. Okay, so they just get old and die. Okay, so in the solid organ transplant patients, what I'm going to just maybe talk about this is yeah, so for CMV disease in solid organ transplant, and as I said, the highest risk for CMV disease is if you've got a donor seropositive and the recipient seronegative, because the recipient has no immune response. But there are also cases where you have donor positive and recipient positive, and you can still get a CMV reactivation and CMV disease in the immunosuppressed organ transplant recipient. So one of the questions is the CTL response important? And so there have been a number of studies looking at whether you've got a higher CTL response against these immunodominant antigens, i.e. 1 and PP65, in patients who have and who don't have CMV disease. So perhaps this is the more recent one where it was a prospective stratification, so it's prospective study, a little bit of a higher recruitment, and it's only in this specific donor positive, receiver positive population. So the patients who had a CTL response against IE1, they had a lower CMV reactivation rate than the patients who didn't have CTL response against IE1. So it's the CTL and T cells against this immediate early antigen that are important for controlling the reactivations, not PP65 specific CTL. And that's because when the latently infected cells reactivate, this is the gene that's expressed first. It's not a virus particle with PP65 in it that's coming in and infecting a new cell. So to control reactivation, it's the CTL response against this antigen which is important. Now, another thing is that, as I said, a lot of or if you're seropositive, then a lot of your T cell repertoire will be dedicated to CMV specific T cells in adults. And so this is something that has actually a big impact on your the profile of your whole immune system. So this is a big study from the Pasteur Institute, the Milieu Antérieur. So they measure a whole bunch of things in healthy donors, and they are looking for genetic and non-genetic factors on the kind of normal immune system. One of the surprising things is CMV serostatus has a massive effect, okay, especially on these T cell subsets. You've got a much higher proportion of CD4 and CD8 TEMRA cells if you're CMV seropositive. Functionally, do we care? A couple of studies coming from Francesco Nicoli and working with Victor Appé in Bordeaux on this. They're interested in senescence in the immune system as you get older. And this is an interesting study here. They looked at the immune response to tick-borne encephalitis virus vaccines in older patients by their CMV serostatus. Why is this interesting? Okay, none of these people had been infected by tick-borne encephalitis virus, right? So you're sure that we have a primary immune response. That's why it's a clever study, okay? A bunch of other things, influenza in 70-year-old people, everybody's been infected. dozens of times. So this is a primary immune response. In the CMV positive group, they had a lower T cell response and a lower humoral response. And they just published a review article that goes into more detail about this. So they are definitely convinced that as you get older, if you're CMV seropositive, the space that this CMV specific response takes up in your T cell repertoire is actually going to make you respond less well to other vaccinations and perhaps infections. So there's a kind of like impact of CMV on our immune system. Okay, I'm not going to talk about gamma delta and NK cells. Just bear in mind that there's a big gamma delta T cell response against CMV, especially in organ transplant recipients. I'm not a big fan of these unconventional T cells. Okay, just maybe one last thing to talk about. If you give me 5 more minutes, give me 5 more minutes. Yeah, thank you. Clotilde, will you give me 5 more minutes? Yes, thank you. Okay. So antibodies. So there's no real evidence for superinfection in in herpes virus, in human herpes virus infections. So I think people think that the humoral response probably is protective against primary infection. Okay, but it doesn't have any impact really on reactivations. Okay, so why is that? Well, for EBV, you've just got the proliferation of the infected B cells. For varicella zoster virus, seems like the T cell response is more important than actually the antibody response. And for herpes simplex and CMV, then you've got cell-to-cell spread, which is very important. So, which means that the humoral response is not really perhaps going to be as effective. But there are some indications from vaccination in CMV or experimental vaccines that, maybe you can have some kind of effect here. So particularly this last one here is that the recombinant, experimental recombinant vaccine can induce antibodies that block cell-to-cell spread that aren't really produced during natural infection. So some ideas that, okay, maybe we can get some vaccine antibody responses that could be effective against reactivation. However, the real effort in understanding herpes virus antibodies and vaccination is really to prevent primary infections. Okay, either in CMV seronegative women, and then you'd be able to vaccinate them, and they would not transmit the virus to their fetuses, or in organ transplant recipients who are especially in this high risk group. If they're seronegative, you could vaccinate them, then you could perhaps protect them from CMV disease. So over the last, 15 or so years, there's been quite a bit of an effort to develop CMV vaccines. So the different things that have been tried are recombinant GB protein trimer. So GB is a surface glycoprotein. I think it's essential for membrane fusion. Well, I don't know. Important for infecting certain types of cells. So there was a big interest maybe when this was published, 2009, had about 50% efficacy. So the trial design here is it's a phase 2 efficacy study. You randomize the women to get the vaccine or no vaccine. You follow up over, so this is like 3.5 years follow up, and you count the number of CMV antigenemia in the both groups, or CMV seroconversion against the other antigens in both groups. So, there were half as many infections in the vaccine group as in the control group. So this was the first ever positive result. So since then, things have been like less encouraging because there's an attenuated virus that was developed. went into a phase 2B, about 700 women in each group, 42% efficacy, just published 2 years ago. So it's going down. In fact, the follow-up studies with this, more like 40% efficacy with accruing more women. 42%, not so good, didn't get on to phase 3. And the last bit of news on this one is what about mRNA vaccines? That's the big new thing in vaccinology, right? So everybody should be making this. One of the problems with the GB is it's not the only glycoprotein that's important for infection. The other big one that's important is the pentamer. That's very, very difficult to express and purify, okay, because it's one copy of 5 different proteins, and they all have to go together. So as a recombinant protein, this one is impossible to make, basically. So this is why mRNA vaccine, maybe it will work. And the announcement hasn't been published yet, but the company, so this was Moderna doing this one in this trial, 7,500 women randomized vaccine and no vaccine group, even worse, okay? So they've announced that they're going to abandon development. So this one fails in the phase 3. Okay, so this is the summary slide. And so for each one, I'm not going to go through this again. It's just what I said. So for each one, I've just tried to give you the main points. How does each virus avoid the innate immune response, humoral response, CTL response? Okay, and what are the main challenges left here? Okay, for HPV, therapeutic vaccine immunotherapy, we need it. HPV, therapeutic vaccine to try and cure chronic hepatitis B infection. For these herpes viruses, CMV vaccine still a big challenge. Okay, so thank you very much for being so kind to me and letting me drone on for so long. And I'd be happy to answer any questions if I know the answers. Otherwise, I'll be very embarrassed.